For a long time, people have had a simple, practical knowledge of genetics. They have known, for example, how to use trial and error selective breeding to develop many varieties of dogs. They know that if a Basset mates with a Basset, the offspring will be Bassets. They know that if a Labrador is mated with a Labrador, the offspring will be Labradors. And if by chance a Labrador mates with a Basset, the result is likely to be something else. Until about 100 years ago, nobody knew exactly how this process worked. But through the work of such scientists as Gregor Mendel and Thomas Hunt Morgan, we now know a great deal. Some of this information must still be described as theory, but much of it can be verified, or at least strongly supported, by mathematical exercise and laboratory experiment. We now know that the patterns of inheritance, the genetic instructions for each physical trait, are passed on to each individual at the time the organism is conceived. What's more, we know that all these instructions are contained within almost all the body cells of the individual organism, specifically within the cell nucleus. To understand the patterns of inheritance, we have to understand something of how the cell nucleus is structured and how it functions. The instructions necessary to make each organism what it is are stored in long thread-like structures in the cell nucleus. These structures are called chromosomes, from Greek words meaning colored bodies, since they absorb color stain readily. Ordinarily, they are too fine to see, but at certain stages of development they become thick enough to be viewed under a microscope. The body cells of each species of organism have a specific number of chromosomes. Human beings have 46. These chromosomes occur in pairs, 23 of them in humans. They are called homologous pairs because each member of the pair is just like the other in size and shape and affects the same traits. Each body cell of the fruit fly, commonly used in genetic experiments, has 4 homologous pairs, or 8 chromosomes in all. In this simplified diagram, the 2 members of each pair are shown next to each other, in 2 shades of the same color. Throughout the life of an organism, its body cells divide for growth and maintenance. In this kind of simple cell division, called mitosis, 2 cells are formed out of 1. Each new cell contains a full set of chromosomes and thus has all the instructions necessary for it to function. Some very simple organisms, like these one-celled animals or protozoa, reproduce primarily by mitosis. However, most organisms reproduce sexually, and for sexual reproduction, special cells different from body cells must be formed. These special cells are formed by a special kind of cell division called meiosis. Unfortunately, the name is very much like mitosis, but the process is different. Both mitosis and meiosis are too complex to describe here in detail, but it is important to understand the difference in their results. In mitosis, the parent cell divides once and simply duplicates itself. Each of the 2 so-called daughter cells has all the chromosomes of the parent cell. In meiosis, the parent cell divides twice and produces 4 gametes. These gametes do not have all the chromosomes of the parent cell. Instead, each contains half the number of chromosomes in the parent cell and 1 member of each homologous pair. All 4 of the gametes produced by the male survive. They are called sperm. 3 of the gametes produced by the female disintegrate, and the single remaining gamete is called an egg. Male and female are commonly identified by the symbols shown here. In the process of fertilization, a male gamete, the sperm, unites with a female gamete, the egg. Remember that sperm and egg each possess 1 chromosome from each homologous pair of each parent. The nuclei of the 2 gametes join, to form the fertilized egg. It contains one chromosome of each homologous pair from each parent. Thus, the original number of chromosomes, in this case 8, is restored. And the fertilized egg contains all the information it needs to divide by mitosis and to develop from an embryo to an adult. You can reinforce part of what you have just seen by a short review. Supply the missing words in these sentences. Please stop the audio for the exercise. Here's how the sentences in the preceding exercise should look. Each of these is called a chromosome. Together, they are called a homologous pair. In each pair, one member comes from the male parent, the other from the female parent. As mentioned earlier, humans have 23 pairs of chromosomes, but obviously each individual has more than 23 traits. Many thousands of instructions are needed for all of an individual's characteristics, from the largest and most obvious to the smallest and most subtle. Each chromosome must therefore contain at least 1,000 of these instructions. A single instruction, a single piece of genetic information, is called a gene. It is now believed that each gene carries a code for the creation of a specific protein substance. A gene may function by itself to control a specific trait, or it may interact with several other genes. In any event, a gene is too small to see and is indicated by the single colored bar on this simplified model chromosome. Genes occur in a fixed linear order on the chromosome. Thus, each gene has a specific locus, from the Latin word for place. Genes ordinarily occur in pairs, each occupying the same locus on each member of a homologous pair of chromosomes. Thus, one gene comes from the male parent and the other from the female. Such genes are often called alleles, from a Greek word meaning each other, because they function together with each other to determine specific traits. Some pairs of genes, or alleles, control a single observable trait. For example, one pair controls whether you can roll your tongue into a U shape, like the person on the left, or whether you cannot, like the person on the right. If both parents have contributed genes for tongue rolling, then you will be able to roll your tongue. On the other hand, if both parents have contributed genes that make tongue rolling impossible, you will not be able to roll your tongue. But suppose one parent contributes a gene that allows you to roll your tongue, and the other parent contributes a gene that makes tongue rolling impossible. If this happens, you will still be able to roll your tongue. How is this possible? When the genes, or alleles, differ, often only one of them will be expressed in the offspring, and the other will not. The gene that is expressed is said to be dominant. The gene that isn't expressed is called recessive. In humans, the gene that allows tongue rolling is dominant. The gene that makes tongue rolling impossible is recessive. If the genes controlling a single trait are the same, the offspring is said to be homozygous for that trait. If, for example, both genes are dominant, the dominant trait, such as tongue rolling, will be expressed. Likewise, if both genes are recessive, the offspring is still homozygous, and the recessive trait, such as the inability to roll the tongue, will be expressed. But if the genes for a single trait differ, that is, if one is dominant and the other recessive, the offspring is said to be heterozygous for that trait, and the dominant trait will be expressed. There are a couple of other terms worth knowing. First, the expressions of inherited traits are called phenotypes. The word phenotype, like the word phenomenon, comes from the Greek word meaning to show or appear. Phenotypes are what appear in the offspring. Second, the combinations of genes that cause the traits to appear are called genotypes. A genotype may be homozygous, in which case the genes are either both dominant or both recessive, or it may be heterozygous, in which case one gene is usually dominant and the other recessive. Here is an exercise for review. A single pair of genes determines whether you are right-handed or left-handed. The gene for right-handedness is dominant. The gene for left-handedness is recessive. What phenotypes will the following genotypes produce? Please stop the audio if you wish to do the exercise. Here are the answers for the preceding exercise. 1. The genotype is homozygous. Both genes are recessive, so the phenotype is left-handed. 2. The genotype is heterozygous. One gene is recessive and the other dominant, so the phenotype is right-handed. 3. The genotype is homozygous. Both genes are dominant, so the phenotype is right-handed. 4. The genotype is heterozygous. One gene is dominant, the other recessive. The phenotype is right-handed. Some phenotypes are like those we have discussed. That is, they are controlled by a single pair of genes, or alleles, which may be both dominant, both recessive, or one dominant and one recessive. For example, the skin color of watermelons, solid green or striped, is controlled in this way. So is whether or not you have a hitchhiker's thumb. Here is an exception. In some cases, neither of the genes is dominant. Instead, they are said to be codominant. If the offspring is heterozygous for the trait, the phenotype will be an intermediate form between the traits of the parents. For example, in horses, the gene for a cream-colored coat is codominant with that for a sorrel. If a horse inherits one of each gene, its color will be palomino, which is a mixture of cream and sorrel hairs. In some cases, 3 or more genes, or alleles, are involved, and the combination of any 2 of them will determine what trait is expressed. For example, there are 3 such genes that determine which blood type you will have: A, B, AB, or O. There is one gene for A, another for B, and a third for O. For clarity here, we will label them that way. Professional scientists use a somewhat more complicated system. A and B are dominant, O is recessive. One genotype for type A blood is homozygous, composed of 2 A genes. One genotype for type B blood is also homozygous, composed of 2 B genes. The genotype for type AB blood is heterozygous, 1 A and 1 B gene. A and B are codominant, and both are expressed in the phenotype. However, since the A and B genes are both dominant and the O gene is recessive, the heterozygous genotype A plus O will produce as a phenotype type A blood. Likewise, the heterozygous genotype B plus O will produce type B blood. The genotype for type O blood must be homozygous and composed of a pair of O genes. That is, both genes must be recessive for the recessive phenotype to appear. Blood types can be identified in a simple laboratory experiment. Here is the equipment needed: a glass slide marked at the ends A and B, a dropper bottle each of anti-A and anti-B serum, a sterile lancet, sterile pads of gauze or cotton soaked in alcohol, and some clean toothpicks. The first step is to put a drop of each serum on the slide. Next, the finger is wiped with a sterile pad and pricked with the lancet.
Ouch! Oh, that didn't hurt. Yes, it did.
The first drop of blood is wiped away with a pad. The second drop is picked up on a toothpick and stirred into the anti-A serum. The next drop is picked up with another toothpick and stirred into the anti-B serum. In each drop of blood mixed with serum, one of two things will happen. The blood may simply dissolve, that is, there is no reaction. Or small granular particles may appear. If this happens, it means there is a reaction between the serum and the red blood cells, causing the cells to clump together. Here are the possible combinations that can appear. 1. A may be grainy and B may show no reaction. 2. A may show no reaction and B may be grainy. 3. Both A and B may be grainy. 4. There may be no reaction in either A or B. If the first combination appears, the blood type is A. If the second, the type is B. If the third, the type is AB. If the fourth, the type is O. Try the following review exercise. If the blood type, that is the phenotype, is A, B, AB, or O, what are the possible pairs of genes, that is the genotypes, that can produce it? Please stop the audio if you wish to do the exercise. Here are the answers to the preceding exercise. Type A can be produced by a homozygous pair of A genes or a heterozygous pair made up of a dominant A and a recessive O. Type B can be produced by a homozygous pair of B genes or a heterozygous pair made up of a dominant B and a recessive O. AB is produced only by the heterozygous combination of codominant A and B. O is produced only by a homozygous pair of recessive O genes. Remember, in human cells there are 23 pairs of homologous chromosomes. In 22 of these pairs, the two chromosomes are very much alike. One pair, however, the one outlined here, is unusual. This pair may consist of two chromosomes that look alike, as at the left, or two chromosomes that are very different from each other, as at the right. These are the sex chromosomes. If the two sex chromosomes are alike, the genotype is called XX, and the person is a female. If the sex chromosomes are different, the genotype is XY, and the person is a male. The same combinations of sex chromosomes produce males or females in many other organisms, such as our little friends, the fruit flies. Now, certain genes may be carried only on one or the other of the sex chromosomes. Such genes are said to be sex-linked. However, the Y chromosome is generally smaller than the X chromosome and contains many fewer genes. Therefore, most sex-linked genes are located on the X chromosome and are more accurately described as X-linked. Some X-linked genes may produce phenotypes in one sex or the other, but not both. For example, tortoiseshell cats are almost always female. To account for this, we must know how coat color is inherited in cats. In cats, there is one gene for black. It occurs only on the X chromosome, never on the smaller Y. So here are the possible genotypes for black. In a female, a pair of genes for black on the two X chromosomes. In a male, a gene for black on the X chromosome and no gene for color on the Y chromosome. There is another gene for orange. It appears at the same locus and only on the X chromosome. So here are the genotypes for orange. In a female, a pair of genes for orange on the two X chromosomes. In a male, a gene for orange on the X chromosome and no gene for color on the Y chromosome. These two genes are codominant. When both are present, the genotype produces a mixture of black and orange, the coat called tortoiseshell. That is, there is a heterozygous pair made up of one gene for black and one for orange. But since each gene can only be on an X chromosome, the cat must be female. Since male cats have only one X chromosome, the genotypes can be one gene, either for black or for orange, but not for both together. The phenotypes can be black or orange, but not tortoiseshell. What have we here? Is it? It is a genotype for a male tortoiseshell. Rarely, very rarely, you may see one. It has two X chromosomes, one carrying the gene for black and the other the gene for orange. This is, of course, abnormal. The extra X chromosome results from a rare error in the process of meiosis. Try this exercise to review what you have just seen. Please stop the audio if you wish to do the exercise. Here are the answers to the preceding exercise concerning Tabitha's kittens. 1. The black male has one X chromosome and one Y. The genotype is one gene for black on the X chromosome and no gene for color on the Y. 2. The orange female has two X chromosomes and no Y. The genotype is one gene for orange on each chromosome. 3. The tortoiseshell is female. She has two X chromosomes and no Y. The genotype is one gene for black on one chromosome and one gene for orange on the other. There are many X-linked genes in humans. You can easily investigate the existence of one of them. Most people see a traffic light as shining either red or green. However, some people will see no difference between the two, except a difference in brightness. These individuals are said to be colorblind for red and green. A simple test to detect color blindness has been devised. Most people who look at a chart like this will clearly see the number shown on the left. Those who are colorblind will see only a random pattern of dots, as on the right. Now, the gene for normal color vision is dominant. The gene for color blindness is recessive. And each of these genes is X-linked. It can occur only on an X chromosome. So here are the three genotypes for normal color vision. The first is a homozygous pair of dominant genes. The second is a heterozygous pair, one dominant, one recessive. Both of these require two X chromosomes and so are female. The other possible combination is a single dominant gene on the X chromosome and no corresponding gene on the Y. It is male. Here are the two genotypes for color blindness. The first is a homozygous pair of recessive genes. It requires two X chromosomes and so is female. The other combination is a single recessive gene on the X chromosome and no corresponding gene on the Y. It is male. Only a small percentage of people are colorblind, and many more of these are men than women. Why this is so will be introduced in the next part of the program. Meanwhile, if you wish to try the color blindness test, here is another chart. The Davises have 4 fine sons, of whom they are very proud. But they always wanted a daughter, too. When Mrs. Davis announced there was a new baby on the way, the family was delighted. Surely this time the baby would be a girl. So the Davises were surprised, to say the least, when the baby turned out to be still another boy. How could it happen? Didn't it go against the law of averages? Well, not exactly. Every child born has a roughly equal chance of being a boy or a girl, and a succession of 5 boys is less likely than some mixture of boys and girls. But only in a very large population will the law of probability result in approximately equal numbers of males and females. To understand the patterns of inheritance, it is necessary to understand something about the mathematics of chance and probability. A simple way of doing so is to use the familiar model of tossing a coin. A coin has only 2 sides, head and tail. Thus, when you toss it, there is an equal chance of getting a head or a tail. But you must toss the coin many times before it becomes probable that you will get close to an equal number of heads and tails. You could try this exercise in a class or other group. The more people involved, the clearer the results will be. First, each person should toss a coin 10 times, mark down each result, and add up the totals for heads and tails, as shown on this sample. Next, find the person with the highest ratio of heads to tails and also the person with the highest ratio of tails to heads. Record these individual ratios in the blanks at the top. Then, divide the whole group into small groups of about 5 people each. Each small group should add up all its scores for heads and for tails, and these totals should be divided by the number of people in each group to arrive at averages. Record these averages in the boxes at the middle. Finally, add up all the individual scores for the whole large group and divide by the number of people in the group to arrive at an overall average ratio. Record this ratio in the blanks at the bottom. Please stop the audio if you wish to do the exercise. Here is how the results of the preceding exercise might look. Among individuals, there will be a wide range of ratios. Some people's results may approach all heads and no tails, or all tails and no heads. The average ratios for small groups of 5 will vary less widely. The average scores for the group as a whole will come closer still to an equal ratio of 5 heads to 5 tails. However, it is unlikely that the overall average will come out at exactly 5 to 5. Here is a graph that shows the same results. Notice that the larger the numbers being counted, the closer the actual figures will come to the expected ones. Now, suppose instead of just 1 coin, you toss 2 at the same time. How does this affect your chances? You are now dealing with 4 different possible combinations of heads and tails. You may get both heads, both tails, 1 tail and 1 head, or 1 head and 1 tail. These combinations are shown on the diagram. The combinations can be expressed as ratios. There is 1 chance out of 4 of getting both heads. There are 2 chances out of 4 of getting some combination of a head and a tail, and there is 1 chance out of 4 of getting both tails. The overall ratio for the different possibilities is therefore 1 to 2 to 1. You can again check possibility against probability by a group exercise. Each person should toss 2 coins 10 times. Enter the scores in a marked-off square like this. Next, find the range of individual scores, the person throwing the fewest pairs of heads and the most, the fewest head and tail combinations and the most, the fewest pairs of tails and the most. Record these scores in the blanks at the top. Then, form small groups of about 5, add up their scores, and divide the totals by the number of people in each group to get averages. Record these averages in the boxes at the center. Finally, add up all the individual scores for the whole large group and divide by the number in the group to get 3 overall averages. Record these in the blanks at the bottom. Please stop the audio if you wish to do the exercise. Here is how the results of the exercise might look. Once again, there will be a wide range among individual scores. When the small group scores are added and averaged, there will be less of a range. The range will be still less when the average scores of the entire group are obtained. However, there may be no perfect 1:2:1 ratio, even in the group as a whole. The same principles of chance and probability operate in the patterns of inheritance. Recall from part 1 of this program that during the process of meiosis, each gamete, an egg or a sperm, receives only one member of each pair of homologous chromosomes. Each member of a pair of homologous chromosomes may be compared to the head or the tail of a single coin. And when a male sperm unites with a female egg in fertilization, the chances of getting a specific combination of any two chromosomes in the offspring are exactly the same as when two coins are tossed together. Now, in the first part of this program, it was explained that genes ordinarily occur in pairs. Such genes occupy the same locus or place on each of a pair of homologous chromosomes. These genes function with each other and so are often called alleles. Each of them can be either dominant or recessive. A single pair of genes, or alleles, controls the shape of the human earlobe. That is, whether it is attached to the cheek or hangs free. The gene for attached earlobes is recessive. If a person has attached earlobes, you can be sure he or she has only recessive genes for the trait. That is, he or she is homozygous recessive. Here, each of the recessive genes is labeled with the small letter E. If a person has earlobes that hang free of the cheek, you know that he or she has at least one dominant gene, but you cannot tell whether the person is homozygous with both genes dominant or heterozygous with one gene dominant and the other recessive. Here, the dominant genes are labeled with the capital letter E. But suppose you know the genotypes, the combinations of genes that produce the phenotypes or visible traits. You then can predict how likely the traits are to appear in the following generation. For example, what if one parent is homozygous recessive for earlobe shape and the other is homozygous dominant? A diagram of the genotype shows the result. All the offspring will be heterozygous. That is, all will have one dominant and one recessive gene. And there will be only one phenotype, the free earlobe, because the gene for it is dominant. Now, suppose two people that are heterozygous for this trait have children. Each parent has one dominant and one recessive gene. This diagram of the genotypes shows the possible results. The chances are 1 out of 4 that the offspring will be homozygous dominant, 2 out of 4 that the offspring will be heterozygous, and 1 out of 4 that the offspring will be homozygous recessive. The overall ratio of genotypes will be 1:2:1, the same as when two coins are tossed. But what will the children look like? What will the phenotypes be? Only a child with both recessive genes will have attached earlobes. Offspring that have both dominant genes, or one dominant and one recessive, will have free earlobes, since only the dominant form of the trait is expressed. Thus, only one child out of 4 is likely to have attached earlobes, while 3 out of 4 are likely to have free earlobes. Here is a similar problem that you can try. 2 of the coat colors in guinea pigs are controlled by a single pair of genes, or alleles. The gene for black is dominant, and the gene for white is recessive. Suppose a female with a black coat is mated with a white male. Suppose also that both the male and the female are homozygous. That is, the female carries 2 dominant genes for color, here labeled with the capital letters CC, and the male carries 2 recessive genes for color, here labeled with the small letters CC. As described earlier, all the offspring resulting from this cross will be heterozygous. That is, they will have one dominant gene and one recessive gene, here labeled capital C and small c. They will all be black because only the dominant gene is expressed. Now, suppose two of these heterozygous offspring are in turn mated. They each carry one dominant and one recessive gene for coat color. What will be the expected results in their offspring? Complete the diagram on the left, placing the genes that the female could contribute vertically along the left edge and those the male could contribute horizontally along the top. Then, fill in the genotypes of the resulting offspring, add them up, and enter the totals in the blanks at the right. Please stop the audio if you wish to do the exercise. Here is how the diagram and the list of genotypes should look when all the blanks are filled in. 1 out of 4 genotypes of the offspring is homozygous and carries both dominant genes. 2 out of 4 are heterozygous, like their parents, and carry one dominant and one recessive gene. And 1 out of 4 is homozygous and carries both recessive genes. The overall ratio of genotypes is the familiar 1 to 2 to 1. But how will such offspring look? What will be their phenotypes? Remember, the phenotype of a homozygous dominant genotype will be black. The phenotype of a heterozygous genotype, carrying one dominant and one recessive gene, will have the dominant color, that is, black. Only a homozygous recessive genotype will result in a white coat. With this knowledge, fill in the outlines of the animals that should be black in these 3 generations. Please stop the audio if you wish to do the exercise. Here is how the diagram in the preceding exercise should look. In the parent generation, one is black and the other is white. In the first generation, all of the offspring are black because they all carry the dominant gene as well as the recessive. In the second generation, 3 out of 4 of the offspring are black and only 1 is white. In other words, in a cross of 2 heterozygous animals, the ratio of possible genotypes is 1 to 2 to 1, but the phenotypes are likely to be 3 black to 1 white. Since genes are too small to see, genetic theories can only be confirmed by examining the phenotypes of organisms used in experiments. Among the most common animals used in genetic experiments are fruit flies. Why fruit flies? These small insects require very little space and are relatively easy to keep. Most important, they multiply very rapidly, so it is possible to obtain fast results. In addition to the normal or wild strain, there are many special strains, which are extremely useful in genetic experiments. One strain has what are called vestigial wings. That is, the wings are shriveled and non-functional. Whether the wings are normal or vestigial is determined by a single pair of genes. The gene for normal wings, here labeled large W, is dominant. The gene for vestigial wings, here labeled small w, is recessive. Incidentally, we are using a simpler set of labels than scientists do. Suppose you have a group of flies with normal wings. That is the phenotype. Now, the genotypes that result in normal wings can either be homozygous, with both genes dominant, or heterozygous, with one gene dominant and the other recessive. Suppose that all these flies have the same genotype. How can you determine what that genotype is? A common experimental method is to take about 5 males with the unknown genotype and cross or mate them with 10 virgin females that are pure recessive. That is, they all have vestigial wings. This is called a test cross or back cross. As soon as the females have laid eggs, usually in just 2 or 3 days, all the parents are removed so they won't get mixed up with the next generation when it hatches. In 10 days to 2 weeks, the adults of the next generation hatch out. They are then anesthetized with ether and spread out under a binocular microscope for examination. Those with vestigial wings are separated from those with normal wings, and both groups are counted. One of two results should appear in the phenotypes. Either all of the offspring will have normal wings, or else about half will have normal wings and half will have vestigial wings. Why is this so? You can explain the results by filling in these diagrams of the genotypes. Remember that the males with the unknown genotype either had two dominant genes, here labeled large W, or else had one dominant and one recessive gene, here labeled large W, small w. Complete the diagrams to find the possible results in the next generation. Please stop the audio if you wish to do the exercise. Here, on the left, is how the first of the two alternative diagrams should look. It was made supposing that each unknown male parent is homozygous and has two dominant genes. A dominant gene is passed on to each of the offspring, and all should have normal wings, because only the dominant gene is expressed. Thus, if all the offspring in the experiment have normal wings, the unknown parents must have been homozygous dominant. Here is how the second of the alternative diagrams should look. It was made supposing that each unknown parent is heterozygous, with one dominant and one recessive gene. Half of the offspring are also heterozygous, but the other half are homozygous, with both genes recessive, and should have vestigial wings. Thus, if about half the offspring in the experiment have vestigial wings, the unknown parents must have been heterozygous. The same kinds of rules apply when you study the patterns of inheritance for more than one pair of genes at once. Ornamental or Indian corn is commonly used for this purpose. Each kernel on the ear is a separate offspring, so each ear provides a sizable population to work with. There is, for example, a pair of genes or alleles that determines whether the color of the kernels will be dark purple or pale yellow. The purple color is dominant, and so will be labeled with a large C. Yellow is recessive, and so will be labeled small c. Another pair of genes on another pair of chromosomes determines whether the kernels will have a smooth surface texture or a wrinkled texture. The smooth texture is dominant, and so is labeled large T. Wrinkled is recessive, and so is labeled small t. Again, scientists use a more complex labeling system than this one. Suppose that a plant that carries both pairs of dominant genes is experimentally crossed with a plant that carries both pairs of recessive genes. What will be the results? First, you have to figure out the possible combinations of genes contained in the gametes, the sperm or egg cells produced by each parent. Remember, a gamete has only one member of each homologous pair of chromosomes. In this case, the parent carrying the two pairs of dominant genes will produce gametes that all contain one dominant gene for purple color and one for smooth texture. The other parent, carrying the two pairs of recessive genes, will produce gametes that all contain one recessive gene for yellow color and one for wrinkled texture. So in the first generation, called the F1 generation, these two kinds of gametes join to produce offspring that are all alike. Their genotype is heterozygous, with one dominant and one recessive gene for each of the two traits. The phenotype will be just like the dominant parent. All the offspring will be purple and smooth. But suppose that plants from this generation are then crossed among themselves. The gametes of each parent now are more varied, as this diagram shows. Each parent from the F1 generation can produce gametes that contain 4 different combinations of dominant and recessive genes. And all of these combinations must be included when diagramming the results of the cross. Here is the diagram. The 4 different gametes from each parent appear across the top and down the left side. 16 squares are required to show the possible combinations of genes in the offspring. There are 9 different combinations if you take the trouble to count them up. Please note one very important thing. Whenever there is a pair containing 1 dominant and 1 recessive gene, the dominant gene is entered to the left of the recessive one. Here's why. If you take only the left gene in each pair, the results will show how the genes are expressed, that is, the phenotypes. 9 out of 16 are likely to show both dominant traits, purple and smooth. 3 would show the dominant color and the recessive texture, purple and wrinkled. 3 would show the recessive color and the dominant texture, yellow and smooth, and 1 would show both recessive traits, yellow and wrinkled. These results can be verified by experiment. Approximately the same ratio of phenotypes, 9 to 3 to 3 to 1, should be found if you count the kernels on ears of such a cross. And the more kernels on the more ears you count, the closer the actual figures should come to the predicted ones. For review, you can try this exercise. What are the possible genotypes and phenotypes of corn that result when a plant that is heterozygous for both traits is crossed with a plant that is homozygous recessive for both traits? To make this task a little easier, the possible gametes of each parent have already been placed on the diagram. Please stop the audio if you wish to do the exercise. Here are the results of the preceding exercise. All the possible genotypes are filled in, with the dominant genes entered first. Then, the combinations of the first genes in each pair are counted up to show the phenotypes that are likely to appear. There will be 4 different ones, and all would be equal. This, too, can be verified by experiment. Crossing a plant that has both dominant and recessive genes for both traits with a plant that has recessive genes only should produce about an equal ratio of purple and smooth to purple and wrinkled to yellow and smooth to yellow and wrinkled. In the first part of this program, we spoke of certain traits such as tortoiseshell color in cats and color blindness in humans. These are produced by genes that lie on one of the chromosomes that determine sex, the so-called X chromosome. The pattern of inheritance of X-linked genes is somewhat different from the others we have discussed. The fertile fruit fly again provides a convenient example. There is a special strain of flies that has white eyes rather than the normal red. Most of these flies are males, which is a clue that the trait is X-linked. The gene for eye color is in fact located only on the X chromosome. The gene for normal eye color, here labeled large E, is dominant. The gene for white eye color, labeled small e, is recessive. Here are the genotypes for red eyes. In a female with 2 X chromosomes, red eyes will be produced either if both genes are dominant or if one is dominant and the other recessive. In a male, the genotype is 1 dominant gene on the X chromosome and no gene at all for eye color on the smaller Y chromosome. And here are the genotypes for white eyes. In a female, white eyes will be produced only if both genes are recessive. In a male, the genotype is 1 recessive gene on the X chromosome and no gene for the trait on the Y. The ratios of white-eyed males and females to normal or wild red-eyed flies can be established by laboratory experiment. The first step is to mate about 6 white-eyed males to 6 red-eyed females that are known to be homozygous for the trait. When the first, or F1 generation emerges, all the offspring, male and female, will have red eyes. This diagram of the genotype shows why. When a female possessing 2 dominant genes is crossed with a male possessing a single recessive gene, the first, or F1 generation, will be about half females with red eyes, each carrying 1 dominant and 1 recessive gene, and about half males with red eyes, carrying only the dominant gene. The next step is to cross male and female offspring from the first generation. When the adults of the second, or F2 generation, emerge, about 1 in 4 are likely to have white eyes. All of these will be males. Again, a diagram of the genotypes shows why. The genotype of each female parent is 1 gene for red eyes on one X chromosome, 1 for white on the other. The genotype of each male parent is 1 gene for red on the X chromosome, no gene for color on the Y. Crossing them produces the following. 1 out of 4 offspring is likely to be a female with 2 dominant genes, and 1 is likely to be a female with 1 dominant and 1 recessive gene. Both of these female genotypes produce red eyes. 1 out of 4 is likely to be a male with a single dominant gene and red eyes, and 1 is likely to be a male with a single recessive gene. It will have white eyes. How is a white-eyed female produced? One way is to cross a female that has both a dominant and a recessive gene with a white-eyed male, which of course has only a single recessive gene. The results are as follows. 1 out of 4 is likely to be a red-eyed female with 1 dominant and 1 recessive gene. 1 is likely to be a red-eyed male with a single dominant gene. 1 is likely to be a white-eyed male with a single recessive gene. And 1 is likely to be a white-eyed female with both genes recessive. Here is one final problem to try. Suppose you have a red-eyed female, but you do not know her genotype. The 2 possible genotypes are either 2 dominant genes for eye color or 1 dominant and 1 recessive. To find out her genotype, she could be mated with a white-eyed male. Fill out these 2 diagrams to arrive at the 2 alternative results. Please stop the audio if you wish to do the exercise. Here are the results of the preceding exercise. If all the offspring turn out to have red eyes, the genotype of the female parent must have been 2 dominant genes. But if about half, or indeed any, of the offspring have white eyes, then the genotype of the female parent must have been 1 dominant gene and the other recessive.